Hello everyone. So today in the class, we will learn about how cells communicate with each other. We have billions of cells in our body, and many of you are thinking how one cell communicate with other neighboring cells, or how, for example, each cells communicate to each other and for the different type of functions which you learned in the previous class. For example, if you think about this COVID situation right now you can imagine that once our body gets the virus and some cells of our body, they pick the virus, then how these cells will tell the other cells that they have got the virus, or what kind of danger signals they will tell to the other cells to alert them, right? These are the various questions, right? So for example, from long time you may be thinking about this fight and flight model, which we also will learn in a few slides. So how cells communicate with each other, what kind of mechanisms are there, this is what we are going to learn in this class. So the learning objective of this particular lecture is that first, what kind of signals are there and how one cells communicate to each other. Cells use chemical signals for their communication. For example, if you have these two cells, they are far apart, and one imagine as a talking cells or transmitting cells, and another is the listening cells, or you can think it a target cells. Target because the other talking cells want to tell something to the target cell, right? So how this talking cell will tell the target cells, how it will send the signals, and these signals they send in the form of chemicals, and these are called chemical signals. And the mechanism which is used to transmit these signals is known as signal transduction pathway. So how this chemical signal will transmit the signal from the talking cells to the listening cells, it will happen in multi-step process, and that multi-step process is called signal transduction pathway. Why do cells communicate? For example, as I mentioned, this good example of impala, of fight or flight. And once they are under the attack, then cells, every body cells has to communicate and allow these trillions of the cells in the impala to talk to each other and coordinate their activity. Because right now, under the attack, this impala needs lot of energy to run very, very fast. So body's brain, muscles, and the energy, they all have energy system, they all have to work in coordination so that this impala can escape from this attack. And how it happens, that's what we are going to learn. Cells communicate for many reasons, right? One example, which is very simple example of two different type of cells, we can here take example of the yeast cell, which is single cell organisms. Unlike the multi-cellular organisms like us, for example, human being, cells needs to communicate in a very, very efficient manner, because multiple different type of cells of multiple different type of organ system, they need to communicate each other. Here is the example of yeast. When they decide to mate, they send the signal, and these signals they send in the form of chemicals. So here is the example. So exchange of mating factors. So here is a yeast cell mating type A, and here another is yeast cell mating type alpha. Okay, and here they are now mating. So each cell type secretes a mating factor, and that mating factor that binds to the receptor on the other cell type. So what you see on the cell surface, and they are receptors. So mating type A will secrete A factor, and the receptor for the A factor is present on the yeast cell mating type alpha. And this alpha strain, they will release the alpha factor, and that will bind to the receptor of the type A. And this binding of the factors to the receptor, this induces changes in the cell that lead to their fusion. So this A and alpha cell will fuse once they secrete the factors and they give the signal to the other cell. So now what you have got is the new A-alpha cell. The nucleus of the fused cells includes all the genes from the A as well as alpha. So that is how these two types of yeast mating type A and E cell mating type alpha, they communicate with each other for the mating process. So in cellular messages, as we are learning how do cells talk to each other, they communicate with each other, they coordinate with each other based on the visual and other sensory inputs. So, and based on this visual and other sensory inputs, what happens is our adrenal glands secrete hormones, and these hormones, they trigger the metabolic processes. So when the prey is being hunted by the predator, the muscles perform at the highest level to run faster, because now the sensory inputs, the adrenal gland secreted the hormones. Hormones are basically chemical messenger, you can think. And now one type of organs like adrenal glands, they are now communicating with the muscles. How? Because adrenal glands secreting hormones, hormones are triggering different metabolic responses, and now muscles is performing at the highest level. Heart beats faster because if the heart beat faster, then they carry more oxygen to the muscles. And now breathing is accelerated, and that need more oxygen. So now you can clearly appreciate that adrenal glands, hormones, muscles, heart, and the breathing, the respiratory system, they are all working in a very coordinated manner to perform a task. This is how the cellular messaging works. So there are multiple ways that even single cellular level, the different type of microorganisms which are just single cell, for example bacteria, they also communicate with each other. So signaling is not just unique to the multicellular organism. Here is one good example of these rod-shaped cells. These are soil-dwelling bacteria, and when there's a scarcity of the food, these cells send out a chemical signal, and then there's an aggregation of these bacteria happen. So by sensing their food scarcity, each bacteria sends signal to each other, and this lead to the aggregation. And these cells collectively, they start making the fruiting body. So if you see the fruiting body like this means that there was a scarcity of the food, and the starving cells sent signal to each other and ultimately led to the fruiting bodies. So this is also called quorum sensing. So quorum sensing is a concentration of signaling molecule that allows bacteria to sense local population density in a process that called quorum sensing. If you think about quorum as a English meaning, the literal meaning, that is the smallest number of people who must be present in a meeting in order to make a decision. What are the different signaling mechanisms in unicellular organisms and in the multicellular organisms? Mechanisms by which cells send a signal or cells receive the signals, and how they process the signals and let other cells know about that signal, right? Evolutionary, if you think that these mechanisms in unicellular as well as in multicellular organisms, they are more or less conserved. Same mechanisms are encountered again and again, and many of these mechanisms, they are universal. In multicellular organisms, there are two types of signaling. One is local signaling. Local means that cells, which are next to each other, they will communicate with the neighboring cells. However, second type is distance signaling, which we will learn in the follow-up slides, where the signaling happens at one side, but the effect even can see on the distant locations. So see, you can see in the figure on the top, there are two cells which are attached to each other, and this one cell signals to the other cells via the gap junctions. So the area where they are attached, it is called gap junctions in the animal cells. However, when plant cells attach to each other, they have plasmodesmata. Plasmodesmata is helping the two cells to interact with each other. And gap junctions or plasmodesmata, they are called cell junctions. And both animals and plant cells, they have these cell junctions that allow molecules to pass readily between adjacent cells without crossing the plasma membrane. Okay, and second type of interaction, which is very, very interesting interactions between the two different and neighboring cells via certain receptors. So receptors are the proteins which have a different type of ligand on another cell. So let us say this cell is having certain proteins which act as a ligand for the other cells which have the receptor for this ligand. And here, two cells in an animal may communicate by interaction between molecules protruding from their cell surface. If you remember, in the immunology class, I was telling that a cell, for example, a T cell would have a receptor, for example, for CD3. Or if there is a cell, imagine this one cell, any of this, is infected with COVID-19 virus. Now, after the processing by the phagocytic cells, if you remember, I mentioned phagocytic cells in the immunology class, where phagocytic cells' job was to take up the virus and now phagocytose it. And now, after this, they generally, the cell's surface get slightly changed. Okay, and that change is expressed on the form of the proteins on the cell surface. And now other cells will recognize these cells by this particular protein which is expressed on the cell surface. And that's how that other cells will know that this is a COVID infected cell. I have told you more like a layman language, but the more technical thing is that this one cell you can imagine is the antigen presenting cell, APC, and antigen presenting cells present an antigen, which is, let us say, COVID-19 here. These cells will have a major histocompatibility complex called MHC, and MHC and the COVID antigen, the combined, that antigen complex with the MHC will present on the cell surface, and other neighboring cells, other immune cells in the body will recognize this COVID-19 antigen and MHC complex and recognize that as a COVID infected cells. That's actually happened in the immune interaction or cell-cell recognition. In local signaling, there are two types of other type of signaling. One is called paracrine signaling, where a secreting cells acts on the nearby target cells by secreting molecules of a local regulator. For example, here it can be a growth factor. So here you see one cell which has a lot of secretory vesicles. And imagine in this secretory vesicles, there are growth factors. Growth factors can be certain proteins which are required for the growth of the other cells, and in this case of target cells. Okay, so this cell, secretory cells or secreting cell, it will have stored proteins or growth factors which are stored in the secretory vesicle. When this cell gives the secretory molecules or which are local regulator such as growth factors to the target cells in a nearby target cell, and that is called paracrine. Synaptic signaling you have been studying probably from class 7th or 8th, where a neuron, the electric signals, this trigger when the electric signal is coming into the neuron, and this will trigger the release of the neurotransmitters. And neurotransmitter then diffuse across the synapse and either give the signal to the next neuron or to the final target cells. So a nerve cell release neurotransmitter molecules into a synapse, stimulating the target cells such as, for example, muscles or a nerve cells or the next one. So this is the different examples. So growth factors, where the growth factors will be required. For example, when you got the wound and then wound need to be healed. And for healing of the wound would require more cells to proliferate. And that's the time for more cell proliferation, you require more growth factors. And this kind of signaling help there. Similarly, cancer cells, for example, cancer cells require lot of growth factors for their growth, and so there it will be required. Okay, so scar tissue formation, the growth factors are required, and in the nervous system, you require a lot of the synaptic signal. Another type of signaling is called long distance signaling. And long distance signaling is mainly for the hormones or the endocrine system. You must have studied endocrine system in your, again, 7th or 8th or 9th grade. That endocrine glands, they are throughout the body and they secrete the hormones. So for example, thyroid, for example, gland and other hormone secreting glands, they are at one side and they will release, they also store the hormones in their vesicles. They release these hormones, and these hormones then go via the blood stream to the entire body. And the target cells can be far away from the endocrine cell. So let us imagine a muscle is the target, and if the muscle is far away from the endocrine cells, the hormone will travel in the bloodstream and reach to the target cell, where this hormone will either go inside the cells or will sit on the surface receptor present on the target cells, and then the signaling will allow the target cells to respond against this hormone. So there are specialized endocrine cells. They secrete hormones into the body fluid, often the blood, and hormones reach virtually all body cells, but are bound only by some cells which have the receptor for this particular hormone. So here example like adrenaline made by adrenal glands sitting on the top of your kidney, and but when the adrenaline is secreted by the adrenaline gland which is near the kidney, that makes your heart rate go up and down when you are stressed, indicating that endocrine cells are far away, and that's how the distant signaling or long distance signaling work. So let us learn that how and what is the mechanism by which adrenaline mobilizes the energy. Actually, it was a molecular model of the adrenaline was actually discovered by Mr. Sutherland in 1971, for which he also got the Nobel Prize. So this is a very important slide showing you how the signaling work. What are the stages of the signal? So if you go right from the left, there's number one is the receiving the signal. So imagine, for example, here the signaling molecule, which is a chemical. It comes to the cell. So the entire orange thing or light yellow you are seeing is a cell. And the blue part, think as an extracellular fluid. So here the signaling molecule is coming, sitting on the top of the receptor, which is present on the cell surface. So cells are receiving the signal. So that is reception. That's the first step, which you have been seeing all through the slides, right? Now what happens? Now this receptor relay the signal by a signal transduction pathway. So this transduction of the signal from one stage to the another molecule to the next molecule, this relay molecules are there in a pathway, and ultimately there's an activation of the cellular response in terms of either new synthesis of the new molecule or new protein, or in some kind of response signal. So third is the response, which is in terms of cellular response in terms of proteins or the new response. So overall, there are three stages of signal. I repeat again, this is a cell which has plasma membrane, and on the plasma membrane, what you have a receptor. One signaling molecule is attached to the receptor. This receptor and signal molecule interaction, this induces or is relay molecules in a different signal transduction pathway, and signal goes from one stage to the second and third and fourth, and that's how the signal is relayed, and ultimately it leads to the response. If you think of analogy, it's more like you give the inputs, and the inputs ultimately goes into the this combinatorial long circuit, logic circuits, and this logic circuit you can by analogy call the signal transduction pathway. So input you can think about as receptor and signaling molecule interaction, and output you think as a response. And the circuit, which is this logic circuit, you can think as a signal transduction pathway. So as I said in the previous slide, that signaling molecule first bind to the receptor, which is present on the cell surface. An interaction of these signaling molecule, which act as ligand, with its counterpart, which is receptor, and it is needed for the cellular response. And what are the most important or most common signaling receptors? These signaling receptors, the most common one, which are present on the plasma membrane, and they recognize the ligands, and these receptors are G protein coupled receptor, GPCR. So majority of your drug receptors, most of your drugs, they bind to the GPCR, G protein coupled receptor. Tyrosine kinases, many of you might have heard for cancer drugs, they bind to the tyrosine kinase. Ion channel receptor, so they allow the ions like calcium ion, phosphorus, all these ions to go in, or sodium, potassium, these ions to go in and out of the cells, and these are called ion channel receptors. Just in little bit more detail, what is GPCR? So this is the largest family of cell surface receptor. If you see in this image, what you are seeing is that there is an outside domain and the inside domain. An outside domain is the signaling molecule binding sites, where the signaling molecule, they come and bind to this site. And if you see the intracellular, this is the signaling domain, where a protein called G protein come and bind, and this G protein decides act as a on and off switch. Okay, so this GPCR is a plasma membrane receptor that works with the help of G protein. And what is G protein? G protein acts as an on and off switch. So how it becomes on and how it becomes off? So when GDP is bound to the G protein is inactive. When GTP, which is more energy form, when that binds, it is active. And what you are seeing in this image, there is almost how many segments of this receptor you call? See, 1, 2, 3, 4, 5, 6, 7. So there's total 7 segments of this receptor of the GPCR. Another type of receptor I mentioned, tyrosine kinase receptor. They are also called RTKs, and they also help in the signal transduction pathway. And how they work? So each receptor, what you see here on your left, they are in a monomer. So what you see, the receptor is there, outside domain, where the red, which is depicting the signaling molecule, it will bind. And what you see, alpha helix in the membrane. And then you see lot of tyrosine kinase in the intracellular domain. tyrosine binding site. So many receptor tyrosine kinase, they have the structure which is schematically depicted here. And before the signaling molecule bind, the receptor exists as a monomer. Notice that each had an extracellular ligand binding site, alpha helix spanning the membranes, the transmembrane domain, and an intracellular tail that has contained multiple tyrosines. And as soon as the signaling molecule bind, what happens, what you see here, so, the signaling molecule can be growth factors. For example, in case of cancer cells require a lot of tyrosine kinase activity because they need to proliferate, they need to grow a lot. And once these growth factors bind, they causes two receptor monomers to associate closely with each other, forming a complex known as dimer. And that process is called dimerization. So how RTK works, if an exam it comes, RTK works when the signaling molecule bind to the extracellular ligand binding site of the receptor. Receptor is a monomer, and this induces the dimerization. And that will ultimately induce the final signal transduction pathway. So once the dimer is formed, what happens? This dimerization, it activates the tyrosine kinase region of each monomer, and each tyrosine kinase then adds a phosphate from ATP molecule to a tyrosine on the tail of the other monomer. So what you see here, then this ATP comes, and then ATP turn into ADP, and this phosphate, it basically this phosphate phosphorylate the tyrosine residue in the intracellular domain of the RTKs. And now that the receptor is fully activated after phosphorylation, it is recognized by the relay protein, which I was talking about, is signal transduction pathway, is recognized by the relay protein inside the cell, and each such protein binds to a specific phosphorylated tyrosine and undergoing a resulting change that activates the bound protein and triggers a transduction pathway leading to the cellular response. So here, for example, what you are saying is these are the relay protein, they're inactivated, but once they will bind to the phosphorylated tyrosine, they will become activated, and each binding protein, each relay protein, they are getting activated and ultimately leading to the different type of cellular responses. For many cellular functions, it is very important to maintain the ion balance inside the cell as well as outside the cell. Okay. So, for example, if you see your hospital reports, you will see they measure sometimes calcium, they measure potassium, they measure sodium, and they always tell you, even for the COVID-19 situation, it's very, very important that our cells, all body cells, work optimally. For their optimal function, balance of the ions in the cell and outside the cell is extremely important. And what makes that balance of the ion inside and outside the cells? These ion channel receptors. For sodium, potassium, calcium, all these things are maintained at a proper concentration by these ligand-gated ion channel receptor. How they work? So, for example, this is a plasma membrane. Signaling molecule comes. So that is representing again here signaling molecule. And these ligand-gated channels are generally closed. For example, in this case, ions you see are outside right now. When this binds, ions goes inside the cells, and then once this ion concentration, for example, in this case, rising inside the cell, inducing certain cellular response. And when the ligand is dissociated from the receptor, the gate closes again and ion no longer enter the cell. So ligand-gated ion channel in which the gate remains closed until ligand binds to the receptor. And when the ligand binds to the receptor and the gate opens, specific ions can flow through the channel and rapidly change the concentration of that particular ion inside the cell. And this change may directly affect the activity of cells in many, many different ways. In case of hormones, for example, or steroids, especially the hormones, aldosterone or others, the steroid hormones. The receptors are not present on the plasma membrane, which you have seen in case of the iron-gated channel or RTKs or G protein coupled receptor. What you were observing there, that all the time signaling molecule was binding on the plasma membrane. However, in this particular case of the steroid hormones, the receptor is present inside the cell. So hormones generally penetrate from the plasma membrane directly into the cytosol, and here in the cytosol they find the receptor for the hormone. So first steroid hormone, aldosterone, for example, I have taken, is passes through the plasma membrane. Next, it binds to the its receptor protein in the cytoplasm and activate it. And this complex, then hormone receptor complex, enter into the nucleus and bind to the specific genes. And here, binding of this receptor complex, receptor hormone complex, it induces the transcription, different transcription factor, and ultimately induce the cellular response by inducing the protein expression. I always told you that most of proteins are the functional unit, and most of the responses, ultimate readout is the protein expression or the gene expression, and building the new proteins, new enzymes, and new activities. So once the first step, which is reception, is done by either GPCR, RTKs, ion channel receptor, or by steroid hormone, the next step is the transduction. And here, relay of the signal from the receptor molecule through a cascade of molecule interaction to the target cells in a cell, it happens via these relay molecules. Okay, so relay molecule can be like only one step, or it can be more than one step. Here I have shown the 3 different relay molecules. And so binding this leads to the change in shape of the receptor, and shape changes leads to the cellular responses. So these are basically the way a cell responds. It can be one step, often in multiple steps, and involve multiple relay molecules, which we are going to learn next. So major relay molecules are protein kinases. Kinases here means the enzymes which causes the phosphorylation, which transfer phosphate group from ATP to the substrate protein. Okay. Another relay molecule is phosphatase. So once the function is done, the phosphorylation is generally removed, and to remove the phosphate, you need phosphatase. They remove the phosphate from the phosphorylated protein. So some protein, so this is like a substrate protein or relay molecule, it gets phosphorylated by protein kinases. So kinase is generally the word used for phosphorylation, and phosphatase is removal of the phosphate and bringing the protein in unphosphorylated form. So here in this particular image, you can appreciate the phosphorylation cascade. Phosphorylation cascade, what you are seeing here is, for example, there's inactive kinase, protein kinase 1. It's in the inactive form, cell is not doing much. But once there is an activated relay molecule, now what you see is the phosphorylation happen, and this here is the active protein kinase 1. And if there are more than one kinases which are phosphorylating, then here you see that cascade is happening. Then the second inactive protein kinase comes, it again get phosphorylated into the become active. Then third inactive protein kinase comes, then again it becomes active. And this ultimate protein kinase, which is active now, it makes the inactive protein an active protein. And when I say active protein, I mean that these are the ultimate proteins which causes the cellular response, because protein is the functional unit. One thing you might have noticed here, that there are or maybe a question may be coming in your mind, why you need these multi-steps. Multi-steps are generally needed to make a process very regulated. If you have only one step process which are not regulated properly, to have a good signaling signal transduction by these protein kinases and phosphatase, and we also need a second messenger. What is second messenger? Some of you might have heard cyclic AMP. Where cyclic AMP fits in this signaling transduction pathway. So, or why it is called second messenger? Because first messenger is generally the signaling molecule. For example, take the epinephrine as an example. Epinephrine binds to the receptor, which provide the reception. So first stage is a reception. So here G protein coupled receptor, and this signaling molecule binds to it. Then this, with the help of G protein, this is a GTP. So that means got activated. The signal goes adenylate cyclase enzyme, and here again the ATP phosphorylate and then it becomes the cyclic AMP. And this signal, when it comes to cyclic AMP, this acts as a second messenger, because here it will again go to protein kinases activation of protein kinases, then multiple steps of protein kinases activation, and ultimately leading to the second messenger, cellular responses. So cyclic AMP basically act as second messenger and connect the first signaling to the signal transduction pathway, which is mediated by protein kinases and the phosphatases. Another type of second messenger are either the IP3, which is also named inositol triphosphate, as well as the calcium ion in our signaling transduction pathway. So once the signaling molecule as a first messenger binds, here you see this put a cascade of the phosphorylation, and here after the phosphorylation the second messenger comes as inositol triphosphate, which again induces various proteins various protein activation or those relay protein activation, and ultimately lead to the function. Okay. And then another here also calcium ion also act as sometimes second messenger, because these calcium ion again they will activate various protein and ultimately lead to the cellular response. So at least you should know what are the different second messenger and what is their role. So once the first messenger comes, second messenger, and then the relay molecule, and ultimately cell respond. And cells respond in many different ways, many different types. Either it can give rise to the gene expression. For example, the enzymes will be synthesized, and there will be catalysis of a reaction by an enzyme, or the cell will change its shape, and to change the shape, it needs its cytoskeleton need to be rearranged, or different type of activation of genes, and those genes can be any type, and new synthesis of hormones, new synthesis or the muscles. Muscles need to make more energy, or maybe digestive enzymes are changed, or nasal something is changing for different purposes. Different type of genes would express. So just to summarize, a real cytoplasmic response to a signal. So here, if you see the first step is reception, so binding of epinephrine to a G protein coupled receptor. So think about this epinephrine. This is actually happening, and we took it from your Campbell book. So here you will find the reception is happening when the epinephrine is binding to the GPCR. Then the signal transduction pathway, transduction is second step happening, where the inactive G protein and then making active G protein, and then different relay molecule here are working in tandem. And here what you see, cyclic AMP also you see, and there's many inactive protein kinases change into the active protein kinases, and ultimately there's activation of glycogen phosphorylase, and this activation of glycophosphorylase is changing glycogen into the glucose 1-phosphate and making many glucose molecules. Basically, that's a response of the cell that activates the glycogen phosphorylase, the enzyme which will go to its substrate glycogen and makes a lot of glucose 1-phosphate. That's a response of the cell. That means cell wanted more energy in the form of glucose, and the body already had your glycogen, which is stored food, and this glycogen needed to be changed into the glucose so that body can get more energy, and that is the response of the body. And here is the transduction pathway, how the different relay molecule, they with the help of second messenger, they activated the enzyme which is glycogen phosphorylase, the hydrolysis enzyme, which will make the glycogen break into many glucose 1-phosphate molecule. So that's how exactly a signal. So you saw the reception, you saw the transduction pathway, and then you saw the ultimate response. That's how cell machinery works. Similarly, another image of depicting a very similar phenomena, that growth factor binding to its receptor on the cell surface. Then signal transduction is happening, phosphorylation cascade is happening with the help of second messengers, and then ultimately this signal goes to the DNA or goes to the nucleus, where it activates different transcription factor, and they induces the gene either on or off depending on the requirement, depending on the cellular response. So I told you that signaling pathway is almost required for all different type cellular functions. So now you can imagine, we have discussed about the hormones responses, we discussed about the many different types. For example, cytoskeleton remodeling, we discussed that how cells need to change its shape. We also discussed how ultimately genes are expressed after certain signals. And here, for example, another example of signaling pathways, apoptosis of programmed cell death. For normal homeostasis means normal cell function. Many cells are alive, and then cells die after a certain time point. So every cell has a life, right? A defined life. Some cells live for longer, some cells have very few days, some cells live for months, some cells live for years. So after a particular time, a cell needs to know that that cell particularly died. And what makes the cell death? For example, here in case of your figure 11.19 in your Campbell book, when you read that apoptosis of a human white blood cells, WBCs, which I taught you in your immunology class as well, this is depicted. So on the left is a normal white blood cell, while on the right is a white blood cell. Okay, that is undergoing apoptosis. And the apoptotic cell is shrinking and forming the lobes or blebs, which eventually are shed as membrane bound the cell fragment. So cell death, basically integral part of the normal functioning of multicellular organism. Immune cells that have done their job. So for example, I was telling you all the time that immune cells, once they are done with the job, they will die because they are no more needed. Because if they are not died, what they will do, because they're activated, and they will kill, or they can also cause harm. So in case of COVID, for example, is ultimate example that the immune, sometimes what is happening, the body is unable to see that whether COVID is gone completely or the COVID causes a havoc in the body, especially immune system. So many patients, what you are maybe hearing, they are very serious because our immune system is overactivated. If you read a little bit more about the COVID, you will see there is a cytokine called IL-6, interleukin 6, which is high. And because of high IL-6 levels, people are having cytokine release syndrome, CRS. And that is causing death and not the virus. So overactivation of immune response is also bad. Having a weak immune system is bad. At the same time, having an overactivated immune system is also bad. Everything should be balanced in life. Okay. So with this, that everything should be balanced in life, neither overactivation of the immune system or the signaling, nor the underrepresentation or under signaling. We will stop this class for now and hope you enjoyed it, and good luck. Thank you.
We will have quiz now. So link for the quiz will be shared in the chat box. I'm going to start the timer now.